Hi, my name is Elena and today we start Circle of Immunology lessons. During our lessons, of course, we should talk about leukocytes, main cells of our immune system. There are two types of leukocytes, myeloid line and lymphoid line, and today we will talk about myeloid leukocytes. There are neutrophils, eosinophils, basophils, mast cells, dendritic cells, monocytes, and macrophages. Let's start with neutrophils. So their development is controlled by different cytokines. Most important is GCSF, it means granulocyte corneally stimulating factor. Also, there are some accessory cytokines like GM-CSF, interleukin-3, and interleukin-6. There are 2 to 7 billion of neutrophils in our bloodstream, but when inflammation occurs, cytokines like interleukin-17 or interleukin-23 induce neutrophils division. During their development, they circulate in bloodstream and then they migrate to liver, spleen, and lungs. So how can they know where to migrate? These tissues release different chemokines like leukotriene and interleukin 8. Their migration is a perplexing process, and they should connect with endothelial cells via molecules of adhesion like beta-integrins and P and E selectins. Main aspects that I want to discuss with you is membrane phenotype of neutrophils. It means like proteins on their surface. So their membrane proteins. First is CD13. It is a receptor for some viruses. Also, they have CD14. It is a receptor for LPS, lipopolysaccharide. Also, they have receptors for complement components like CR first, second, third, and fourth. So, next important group is Fc receptors. Fc receptors are receptors for Fc domain of antibody. Fc domain is connected with antigen. It stimulates phagocytosis and cytotoxic activity. There are two types of Fc receptors on neutrophils. It is Fc gamma R second and Fc gamma R third. They are low affinity receptors. We'll discuss types of Fc receptors soon. Also, neutrophils have a receptor for leukotriene B4. It is something like chemokine. They express MHC second, but only after activation, and they express MHC first constantly. Also, they have receptor for GCSF, of course, because it is very important factor for their development and activation. Receptor for GM-CSF, receptor for interleukin 17, 23, 8. All of these cytokines are vital for their development, for their growth and life. Also, they have receptors that names toll-like receptors. We will discuss them in another lesson because it is a very huge group of receptors and they are vital for our innate immune system. Okay, so we discussed the set of receptors on the surface of neutrophils, but what about some specific granules in them? So neutrophils are divided into granulocytes. Yes, there are two types of leukocytes, granulocyte and agranulocytes. Neutrophils are granulocytes. They have set of granules. First type of granules are specific granules. There are some enzymes. For example, lactoferrin, it is enzyme that can take iron from bacteria, so bacteria's ETC stop working and it dies. Also, they have alkaline phosphatase. It can dephosphorylase substrate and regulate their activity. Lysozyme, it is antibacterial substance. BPI protein, it can bind vitamin B12. NADPH oxidase, that can catalyze oxygen explosion and generation of active forms of oxygen. It is the main antibacterial factor of neutrophils. next type of neutrophils granules are eosinophils granules. So these granules contain a wide range of proteins that are active in acid environment. For example, myeloperoxidase form hypochlorite anion. It is antibacterial factor. Also, there are fucosidase, nucleosidase, monosidase, N-acyl, glucosinamidase, lysozyme, neutral proteases like collagenase, catepsin G, and others. Third group of neutrophil granules are gelatinoses granules that contain gelatinase for destruction of basement membrane during their migration to tissues. And the last group is releasing vesicles with alkaline phosphatase. It maintains interactions with environment of neutrophils. And of course, the main thing that we should discuss is the mechanism how neutrophils works. Okay, first cells that encounter a pathogen during its penetration to our body are resident macrophages. They phagocytose this pathogen, and after it they release chemokines to attract different cells to the inflammation place. For example, neutrophils, lymphocytes, and so on. But also they can release a set of cytokines to activate different cells, not only our leukocyte but also endothelium cells, epithelium cells, and so on. So during inflammation, different cytokines stimulate neutrophils. They activate, release different substances, including gelatinases that break our basement membrane and provide migration to the tissue. Also, these cytokines stimulate releasing of adhesion molecules, so it helps neutrophils to migrate to the tissue. They interact with endothelial P and E selectins. Then neutrophil encounter pathogen, phagocytosis, their specific and eosinophilic granules fuse with the phagosome, so they kill bacterium of another pathogen. They are cells of our first line of defense. There are cells of our innate immune system, and all nearest lessons will discuss exactly our innate immunity. So neutrophils also have a specific trait. There is an ability for NETOs. What is it? They can express specific nets. These nets is formed from DNA. So neutrophil release this DNA. Of course, it dies, but bacterium can stuck in this and lose its pathogen characteristics. It is a very important and very interesting fact about neutrophils. Okay, next type of our granulocytes are eosinophils. They make up a much smaller part of our blood cells, about 0.5 to 2%. They circulate in bloodstream less than 18 hours, and then they migrate to our tissue and spend there about 10 to 7 days. So they also have a set of proteins on their surfaces. First is CD9 and CD35. These are receptors for complement components. Also, they have receptors for IgG, Fc gamma R second, and Fc gamma R third. And also, they have receptors for IgE. It is Fc epsilon R2, R second. And they have receptors for cytokines like interleukin 5, interleukin 3, GM, CSF, receptors for different chemokines, for example CCR3. We'll discuss different chemokines soon. And also they express MHC after activation and MHC second after activation, and MHC first constantly. Also, they have adhesion molecules on their surfaces like beta integrins type 1st, 2nd, and 7th. As all granulocytes, they also have granules, specific granules. First class is eosinophilic granules. It is big specific ones that contains, for example, main basic protein MBP and eosinophilic cation protein ECP. These two proteins act against helminths. Also, eosinophils contain two more granules, two more substances in their granule. It is eosinophilic peroxidase EPO and neurotoxin EDN. Also, we can say that ECP and EDN break down RNA molecules. They are RNAs, so they take part in antiviral defense. But all four proteins can be toxic for our organism. Okay, so in the first type of granule, there are these four types of proteins, and also there are some cytokines and enzymes like collagenase, catepsin, elastase, myeloperoxidase, and others. Also, they have little granules with different enzymes like acid phosphatase, aryl sulfatase, peroxidase, and others. Next, third types of the granules are primarily granules. They have charcot-laden crystal protein. And fourth is lipid granule with components for icosanoids. Apart from these molecules on their surfaces, eosinophils release different factors. For example, cytokines like interleukin, all of inflammatory interleukins like 2, 3rd, 4th, 5th, 6th, 8th, 10th, 12th, 3rd, 16, 18. I will write them on the desk. Also, they produce TNF, it is also inflammatory factor. Interferon gamma, we'll discuss it later, it is very important factor. TGF beta, GM-CSF, and different chemokines like eotoxins, arantes, and MIP. But at the same time, they release receptors for chemokines. Also, for chemokines, arantes, for eotoxins, they release CCR first, second, and third. So they should release chemokines and they should react to the chemokines. It is important. So what is eosinophilic function in our body? Primarily, they have an ability to perform extracellular cytolysis. So it is used for killing multicellular parasites like helminths. Also, they take part in antiviral defense, and moreover, they can activate basophils and mast cells, so they have a role in allergy development. Interesting thing, they can induce processes of our adaptive immune system by T cell activation. Okay, next type of our granulocytes are basophils. This type of cells is also few in number in our blood. They circulate in blood, and after activation they migrate to the tissue. They also are vital for TR2 cells development. We'll discuss it. They release interleukin 4, and this cytokine is very important for TR2 cells development, life activation, and so on. By the way, they take part in allergic reactions with mast cells. So, as other cells, basophils also express different proteins on their surfaces, different receptors. Set of receptors on their surfaces. First is a receptor for FMLP. It is very important bacterial peptide. We'll discuss it when we'll talk about our innate immune system substances. Also, they have receptors for anaphylatoxins. These are complements like C3A, C5A. All of them are something like chemokines. They attract different cells and stimulate phagocytosis. Also, they have receptors for alpha and beta chemokines, CC and CXC proteins, chemokines. Also, they express high affinity receptors for IgE. These are FC epsilon R first, and also they express low affinity R second. Most important receptor on the surface is histamine receptor. It is H second, and also they express different types of TLR, toll-like receptors. Yes, they are also very vital for our innate immune system. Okay, very close group are mast cells. Mast cells and basophils have a lot of similar trait, but the difference is next. First of all, mast cells are located in the tissue and they don't circulate. They maintain inflammation. They are major factors during immediate hypersensitivity like allergy. They are rather long-lived cells, about months to years. So their membrane phenotype. Also, they express Fc epsilon receptors for IgE. They express receptors for IgG like Fc gamma receptors. First, it means high affinity. They express CD117. It is a receptor for CSF stem cell factor. And they express receptor CD23. It is a receptor for interleukin 3. Interleukin 3 and stem cell factor are very important for their development and for their life. Also, they express receptors for complement components like C3b, C5b. Thanks to them, opsonization is carried out. Also, they express MHC second. They are APC cells. They can present antigen to other cells. And also, they express MHC first, like all other cells, all other nucleated cells. Also, as APC cells, they release, they express CD86. It is co-stimulation molecule. And also, yes, TLR, toll-like molecules, like all other innate immune system cells. By the way, I think that we should say that there are two types of mast cells. There are two populations. First is mucosal T mast cells. They express tryptase, so they are T mast cells. And also there are connective tissue mast cells, CT mast cells, because they express chemotryptases. Both of them are developed in bone marrow, but T mast cells are dependent on thymus. Growth factor for both of them is CSF stem cell factor, but cofactor for T mast cells are interleukin 3 and 4, but for CT mast cells it is only interleukin 3. Okay, that's all about mast cells, and next group is absolutely vital in our innate immune system, especially when we are talking about first reaction, first line of defense. And we start talking about macrophages. Only part of macrophages are originated from monocytes. Others, like microglia, Kupffer cells, are originated from embryonic tissues like yolk sac. Differentiation from monocytes to macrophages is regulated by MCSF, monocyte colony stimulating factor. So macrophages carry out their main function through different types of receptors. So first type of receptors are TLR, toll-like receptors. You should feel it, how important TLR are in our innate immune system. They can recognize everything, every type of pathogen. These molecules of pathogen calls PAMPs, like pathogen-associated molecules patterns, and so they can recognize everything: bacteria, viruses, fungi, protozoa, multicellular parasites, and everything. They are really significant. And we have two types of them, like TLRs that are located on the surface of the cell and intracellular TLRs. Extracellular, like on the surface, are TLR first, second, fourth, fifth, and sixth. They can recognize some bacteria that is extracellular located. And also we have TLR third, seventh, eighth, and ninth. They can recognize them in our cell. So it is defense from viruses. There is also a helper. It is CD14. It helps TLR to recognize LPS, lipopolysaccharide. Yes, it is like an additional fact. Next group of receptors of our macrophages are lectin receptors. They can recognize some saccharides, especially D-glycosyls like glucose, fructose, galactose, mannose, N-acetyl, glucosamine, and also others. On our own cells, these molecules are shielded by sialic acids, so macrophages attack only pathogen cells, but not our ones. The most important lectin receptor is mannose receptor, and also there are DCSI and dectin-1 and others. Also, macrophages express scavenger receptors. They are very useful for eating apoptotic bodies, yes, after cell death, so we will discuss it in the topic of apoptosis. And also they of course release FC receptors. As for monocytes, they release FC receptors for IgG and even high affinity air first, but also low affinity too. But macrophages can express only low affinity receptors. For IgG, it is Fc gamma R second and R third. Also, both of monocytes and macrophages have receptors for IgA and IgE. And next type of receptors on their surfaces are receptors for complement components. They can recognize opsonized structures, and it can alleviate process of phagocytosis and cytotoxic activity. There are CR first, second, third. All of these receptors are receptors for C3b and C5b. And the last type of receptors on the macrophage surfaces are receptors for anaphylactoxins. These are components of complements like C3a and C5a. I understand that now it is too complex to understand the difference between C3b and C3a, but we will discuss complement system soon. But also on the surface of macrophages, of course, there are other types of molecules like MHC second. They are very good APC. MHC first, co-stimulating molecules like CD80 and CD86, and also they have MCSF receptors and receptors for interferon gamma. They are very important. And receptors for different inflammatory cytokines like interleukin 1, 6, 12, 18, TNF, and so on. If we are talking about macrophages, we should emphasize that their migration, their motion is very important, and it is based on integrin molecules like beta first integrin group and beta second integrin group. Beta first provide connection with extracellular matrix like elastin, collagen, laminin, and so on. We can do it via VLA molecules, VLA2, 6, 4, 5, they are expressed on macrophages a lot. And also we have beta-2 integrins. They provide interaction between cells, between macrophages and endothelium or other cells. For example, there are LFA molecules, LFA receptors, first, second, and there are a lot of them. So they can interact with endothelium, and endothelium express ICOM molecules for these LFA. And also there are third types of integrins. It is SLAX molecules. They are vital too. They connect with endothelium and other cells with their selectins, P and E selectins, on the endothelium surfaces and other cell surfaces too. And the last type of our innate immune system cells, these are dendritic cells. So this group is really diverse. There are a lot of population of dendritic cells. They have different origination, like someone is originated from lymphoid line, others are originated from myeloid line, and there are a lot of groups in these big groups. So the major fact about dendritic cells that they can present antigen epitopes like APC 100 times better than macrophages. So DC dendritic cells are absolutely specialized on this process. It is their occupation. They always present antigen to lymphocytes. They don't kill, they are not so cytotoxic cells. The main function of dendritic cells is to phagocytose, to represent this epitope, and to show it to lymphocyte. Okay, we can divide dendritic cells into two groups. It is classical dendritic cells and plasmacytoid dendritic cells. Let's talk about last one. Plasmacytoid dendritic cells, we also can call them IPC because they are interferon producing cells. It belongs to lymphoid line. IPC are immature cells. They are migrating with help of interleukin-3 and bacterial peptides. They are major source of interferon types. Type first, it is interferon alpha, beta, and omega. Dendritic cells start producing interferons after connection with viruses. After stimulation by a virus, they help the development of T helpers to Th first type, and Th first type produce interferon gamma. But if they get a signal from interleukin-3, they help development T helpers to Th second type, and Th second type of our T cells produce interleukin-4. We'll discuss the difference between Th cells. They are absolutely different, so yes, remember it. Dendritic cells migrate to the tissue despite they are not mature cells. Tissue DC cells can be resident or inflammation types, resident lymphoid dendritic cells, for example, in derma, in epidermis, in intestinal mucosa, in lung surfaces, and so on. Also, we have resident myeloid dendritic cells in thymus, in germinal centers of lymph nodes, in marginal zone of spleen, in liver, and so on. So I think that now we can finish our first lesson about our immune system. If it was interesting for you, I understand that it is rather complex, but if it was interesting for you and you want to hear some new information, please leave comment, and if you want to ask me something, I will be glad to answer. Yes, so please subscribe, leave comment, and I will wait you on the next lesson. Bye bye.